Namaskar everyone. So in this video we will discuss water reducing sugar. Water reducing sugar. Before going to reducing sugar, let's understand what are these sugar. Sugars are those compound which are sweet in taste and they are carbohydrate. Right? As for example, you see our glucose. Glucose is one example of sugar, isn't it? Okay. Now what are reducing sugar? Oxidation reduction you all know, right? Like gain of electron is reduction, is it? Loss of electron is oxidation. So the sugar which can convert Cu2 plus ion into Cu plus ion are called reducing sugar, is it? Cu2 plus ion, right? So that sugar particularly will donate one electron to this Cu plus. Okay, so when the sugar will donate electron to Cu plus, Cu2 plus will accept that and they will convert into Cu plus, right? So that sugar are only called reducing sugar, right? Now question is that, okay, so which kind of sugar they can do their activity? Before going there, we have to understand that two tests we are doing commonly. One is Benedict test and another one is Fehling test. So by Benedict test or Fehling test we can identify reducing sugars, okay, reducing sugar which can convert Cu2 plus into Cu plus, right? Now particularly if you'll see the case of the Benedict test, this Benedict test, in Benedict test we will use copper sulfate. Copper sulfate will mix with water, and when we'll mix with water, this copper sulfate will dissociate into Cu plus SO4 minus. Okay, right? This copper sulfate, this Benedict test, this Benedict reagent, it's a blue color reagent. Okay, it have copper sulfate and it's a blue color reagent, right? You take Benedict reagent, okay, fill in this test tube. Particularly you fill that Benedict reagent in this test tube, okay, like you see I'm filling. After that what we do, we will heat it, okay. After heating we will add some sugar here, okay. After adding sugar, if color of reagent is changed, like from blue to green or yellow or red, that means the sugar which was added is reducing sugar. I am repeating it again, try to understand it. See, in a test tube I taken Benedict reagent mixed with water. Benedict reagent have H2S copper sulfate. That copper sulfate actually, when it will be mixed with water, it will convert into, it will dissociate into Cu2 plus to Cu plus. Okay, then you add some sugar here, unknown sugar you add, that you have to know that this sugar is reducing sugar or not. After adding that particular sugar, you heat that reagent in a test tube, okay. After heating it, if you find that the color is changed, like it's became green color, like it's became yellow color, orange red color, okay, or brick red color, right? That means this sugar is reducing sugar. If color changes, why color changes, see, actually this reducing sugar will convert this Cu2 plus into Cu plus and color of that solution changes, right? So this is what called Benedict test. In Benedict test, by Benedict test easily you can identify by this color change that sugar is reducing sugar or not. Now question is that which kind of sugar, on the basis of biochemistry, on the basis of structure, we can tell which kind of sugar have capacity to do it. So remember student, those sugars which have free aldehyde or ketone group, see here, those sugar which have free aldehyde or ketone group. You all know that carbohydrates are polyhydroxyaldehyde or polyhydroxyketone. That means carbohydrate have either aldehyde group or ketone group. Yes or no? Aldehyde or ketone groups are present or not? Yes. Either aldehyde or ketone groups are present. See, if this aldehyde or ketone group are have engaged in formation of glycosidic bond, then we will not call it free aldehyde or ketone group. But if they have not participated in formation of any kind of glycosidic bond, right, so we will say okay, they have free aldehyde and ketone group. So the sugar which have free aldehyde or ketone group, we will call them reducing sugar. Like all monosaccharide do not form any kind of glycosidic bond. Okay, so all monosaccharides are reducing sugar. Okay, all polysaccharides are non-reducing sugar. Right, now what about our disaccharide? That is interesting, like you see this lactose sugar, milk sugar. Okay, lactose and maltose both are reducing sugar. Sucrose, 3 halos are non-reducing sugar. Sucrose, sucrose present in sugar cane. 3 halos is present in blood of the insects. Fine, so both are non-reducing sugar. Question is that why? Try to understand it. Like I am taking example of, for explanation sake, I am taking example of lactose. Lactose have two kind of sugar, one is galactose, 6 carbon sugar, and another one is glucose. This is also 6 carbon sugar. Both are aldehyde. Fine, like you see, this is galactose, first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon, first, second, third, fourth, fifth, sixth. What happened, this first carbon will react, the first carbon of galactose, obviously it is aldehyde group, first carbon is aldehyde group. Okay, it react with fourth carbon of glucose, right? See, I think you all know, you all have basic idea of the nomenclature of organic compound. So if you have a compound actually, so during nomenclature we will identify numbering of the carbon on the basis of seniority. Okay, so if the sugar is aldose sugar, first carbon will be considered, aldehyde group will be considered as most senior carbon, and that carbon will be considered as first carbon. Okay, so first carbon actually in the cyclic structure, how it's formed, that we have discussed it already, right? I just want to say here, at first carbon aldehyde group present. Obviously in this cyclic structure they temporarily reacted with fifth carbon, but first carbon will be considered as aldehyde group only. So first carbon aldehyde group of galactose reacted with fourth carbon OH group, right? And then they are forming glycosidic bond. Clear, students? They are forming glycosidic bond. Now you tell me here, aldehyde group is got engaged or not, because they are forming beta 1,4 glycosidic bond. So here first carbon engaged or not? Galactose first carbon engaged, but glucose first carbon is free or not? Yeah, glucose first carbon is free. That means they have free aldehyde group. That means this lactose have free aldehyde group, and as they have free aldehyde group, they can convert glucose into, the Benedict reagent. In Benedict reagent, Cu2 plus, they can convert into Cu plus. Okay, so they are reducing sugar. Lactose is reducing sugar. Now you see the case of maltose. Okay, maltose first carbon react with fourth carbon of, in maltose first carbon of one glucose react with fourth carbon of another glucose. Now aldehyde group is present at first carbon, so maybe this glucose is alpha glucose, maybe this glucose first carbon participating in formation of glycosidic bond, and here but second one fourth carbon is participating in formation of glycosidic bond, yes or no? So bond form here glycosidic bond, that is called alpha 1 4 glycosidic bond. We will call it, see, this bond will be called alpha 1 4 glycosidic bond. Okay, that means here first carbon engaged in formation of glycosidic bond, but second glucose first carbon is free or not? Yeah, it's free. You see here, it's free, right? So they will reduce the Cu plus into Cu2 plus into Cu plus, Cu2 plus into Cu plus. Okay, so again our maltose is also reducing sugar, right? Now you see the case of our sucrose. See, sucrose, if you'll see, our sucrose have alpha 1 beta 2 glycosidic bond. What is this alpha 1 beta 2 glycosidic bond? That we will see in the different video, but try to understand. Our sucrose have two compound, one is glucose, another one is fructose. Okay, now you all know that glucose is 6 carbon sugar. Okay, fructose is also 6 carbon sugar, but it's aldose. Okay, and this fructose is ketose. So in this ketone sugar, in this fructose, ketone sugar present at 6th carbon. Sorry, ketone group will be present at 2nd carbon. Is it or not? Yeah, in fructose ketone group present at 2nd carbon. We have discussed in all different video, right? And glucose aldehyde group present at 1st carbon. So this glucose 1st carbon and and fructose 2nd carbon engaged and they form glycosidic bond. Yes or no? They form alpha 1 beta 2 glycosidic bond. That means glucose aldose group is also engaged here and fructose ketone group also engaged here. That's why they form alpha 1 beta 2 glycosidic bond. Yeah. Okay. So sucrose, both aldehyde and sucrose have two sugars and their aldehyde group and ketone group are blocked in formation of glycosidic bond. So they are non-reducing sugar. Remember, sucrose is non-reducing sugar. Now come to 3-halose. 3-halose have 2 glucose. 3-halose have 2 glucose and both glucose are engaged in formation of glycosidic bond. Okay, which glycosidic bond? alpha 1 glycosidic bond. ठीक है, right students? So again our fructose is also non-reducing. Our 3-halose is also non-reducing sugar. Okay, so those sugar which have free aldehyde or ketone group, they can convert Cu2 plus into Cu plus. That's why they are reducing sugar. Okay, so in this video we discussed what is reducing sugar. In this video we also saw that what is Benedict test. Okay, in Benedict test, you see, Benedict test we use to detect glycosuria, glucose in urine. Glycosuria, for detection of glycosuria we use this Benedict test. How? See, those who have diabetes, in diabetes, you know, glucose releases in urine and that condition is called glycosuria. So if the person is suffering from diabetes and glucose is releasing in urine, that means person have issue of glycosuria. Glucose is reducing sugar. Okay, I will take that person urine, okay, mix with Benedict test. After mixing with Benedict test, we'll take urine, right? We will heat that and we will pour 1 drop of Benedict reagent in that. Okay, we taken in this test tube, we taken urine, added 1 drop of Benedict reagent. Okay, if in that person is suffering from diabetes and that have glucose, immediately they will convert that urine into green color if glucose is less than. If glucose is more, then it will be yellow color. If glucose is even more, then it will be orange yellow color. And if glucose is very high, then it is then it will be our brick red color. Okay, fine. So this is what our reducing sugar. Remaining things we'll see in next video.